As usual, we have 3 slams. Some of you are new to this process. The 3 slams, each one is a 10-minute talk, and we are pretty tough with the timer, and then 5 minutes for questions. The 5 minutes for questions are because for the audience, that's invariably the funnest time to be able to ask the questions that you want to ask. And so we'd really like to spread them around to the audience. At the end, there will still be some time that you'll be able to ask questions to people if you just hang around. If you want something to eat or drink in the middle, this is pretty informal. You can get up and grab it. Try not to walk right in front of Eric back there with the camera. And we'll go forward. Any questions before we start? As we go through this process, if you have any suggestions, we're really happy to have them. We have 3 speakers tonight from 3 different colleges that will give you a perspective of some of the exciting things going on in the College of Sciences. The first speaker is Dan Goble, and Dan is from the School of Exercise and Nutrition Sciences in the College of Health and Human Services. Dan's going to tell us about his recent research and how it led to an entrepreneurship opportunity.
Thank you. I'm honored to be here. I'm honored to have been asked to be here. It's pleasure. I see a lot of familiar faces, which is outstanding. So a lot of you, I guess, already know a little bit about this, but I'll try to give you a little more meat than normal. What I'm going to talk about is actually how I accidentally started a medical device company without even trying here at San Diego State. To give you a little bit of my background, I'm a junior faculty member here. I got here about 4 years ago. I have a PhD from Michigan, spent 3 years in Belgium drinking beer, and somehow ended up in the beautiful sunshine, so can't beat that. I'm going to lead into this with some talk about concussions, and you can't talk about concussions without showing one. So here's a horrific concussion of Javid Best from Cal several years ago. If you go to YouTube and look up worst concussion ever, this is actually what comes up. And they're going to show it from about 5 angles. You'll see the linebacker actually vaults him further into the air. He later went on to the NFL, got more concussions, and then had to quit the game because of too many concussions. These are a big deal right now. Why are they a big deal? 2 reasons. One, for medical reasons. So what they're finding with some autopsies of some former NFL players' brains is that people who play these sports a long time don't look normal. So this is what a normal slice of brain looks like. It's nice and white. And then these are some professional athletes who are the exact same age, same kind of demographic characteristics. You can see there's a bunch of brown stuff on the brain tissue. That's plaques and scars, and really what they like to call the new hot term CTE, chronic traumatic encephalitis. Interestingly, these brain slices look a lot like brain slices of people who have other brain disorders like Parkinson's disease, such as Muhammad Ali. The other reason concussions are a big concern is because of money. So there was a huge $700 million lawsuit a couple years ago, former players versus the NFL, and they're talking about shutting down the league in 10 years and all these things. So it's a big deal. So since it's a big deal, how do we actually deal with it? Well, we're putting tons and tons of protocols out there for assessing concussions. What's a typical assessment look like? These are the guidelines that were established in 2012 by a whole bunch of smart athletic training and medical professionals who took a conference in Zurich, Switzerland, and said this is how we need to be assessing concussions. First thing is symptoms. You're going to ask an athlete, are you dizzy? Do you have double vision? Are you sensitive to light? All these symptoms we associate with a concussion. This is the first layer, and it's a terrible layer because it all depends on the athlete's response, and they can lie about it. So they added a second layer to the concussion assessment. That's the cognitive assessment. That's your questions that really are kind of memory questions. What was the score in the game when you left? Where are you at right now? What day of the week is it? A little bit better, but athletes are smart, particularly professional athletes who have a lot at stake. So they actually start ingraining these things in their brain before games. They know the questions by now, and so they'll sit there before a game and go, today is September 22nd, 2015. It's a Tuesday. And they can actually get through those cognitive assessments in some cases. So the third layer to the concussion assessment is a motor assessment, and in particular that motor assessment is a balance test that's recommended, and that's a little bit trickier for athletes to fake. One problem, though, is that this is the recommended balance test that everybody is supposed to use out there according to these guidelines developed in Switzerland. It's called the BEST test for balance error scoring system. And it has 6 conditions where the person's standing with their feet in different configurations to make it harder, so next to each other versus in front of one another on one foot. And also some conditions where you're standing on a spongy foam surface, which also makes the test harder. And what the athletic trainer does is they watch them for 10 seconds in each of these postures, and they try to count errors. Errors are things like, does the hand come off the hip? Does the foot touch the ground? Do the eyes open? Do they step out of place? And that is just a subjective, terrible test, and research has shown pretty convincingly that it's inaccurate, unreliable. If you have 2 trainers try to do this test on the same person, they get different numbers. That's because you can't look at the eyes and the hands and the feet all at once. It's just too challenging. Also, what qualifies as a step out? Does it have to be a huge step out, just a small step out? So why do they actually use this test? The reason they use this test is because it's dirt cheap, and you can have anybody do it. Your Pop Warner football teams can do this, as well as your professional teams. So in typical American fashion, we value quantity over quality and say it's okay, it's much better to do a whole lot of bad testing than it is to do a little bit of good testing. Sorry, I just took a shot at the Americans. Didn't mean to do that. I'm trying to get my green card too. Coincidentally, this is the test that the NFL uses. So even though the NFL can afford to do anything it wants, it actually uses this very cheap test. And in fact, they actually throw out 3 of the conditions and just do the top 3 conditions, which is even more unreliable, testing shows. So what is the best way to measure balance? It's with a force plate. These are force plate devices that already exist in the market. If you had a balance problem, went to a balance clinic, this is how they would measure your balance. They would have you stand on a force plate. They'd measure how much your body sways from the forces, and the idea would be if you swayed less, your balance is better. The problem is they're all very expensive, thousands of dollars, up to over $100,000, which is too expensive for most sports teams. So what do we need? We need an affordable force plate that gives us objective, accurate, reliable results that everybody can get their hands on. So that's what I invented over the last 3 years, and I'm promoting and trying to sell. We call it the Balance Tracking System, or B-Trax. It looks exactly like this. It's light, it's portable, you run a 2-minute test. It doesn't require power, you just plug it into your laptop, powers off your laptop, has user-friendly software. It is amazing. How did we do this? So 3 years is a long time. How did we actually get to this? It's a little bit of a story. I'm going to tell it to you. This is Jasper Chang. He was a master's student in my class when I first got here. One of the things we do in my class, because I had almost no money when I started up here for startup money and equipment, I got really creative and started using cheap alternatives for equipment. One was the Nintendo Wii balance board. It's a $50 balance board, and I hacked into it with some software I created and turned it into the equivalent of one of these expensive force plate devices. It actually was about 99% as good as the $1,000. Is that 7 minutes?
Yeah, 3 more.
3 more minutes, okay. So Jasper was working at UCSD. They were going to buy a very expensive system, but he had taken my class and he said, don't buy the expensive system. I'll ask Dr. Goble to make a system out of these Wii boards. And over the course of a weekend, I made a little bit of software. Next thing you know, they've tested 200 athletes. Then my director said, this is a business. You can't just give this technology away, so I declared it. Got hooked up with the Zahn Center. Kathy Poocher was pretty invaluable in all this process. They got me hooked up with the pieces I needed to start a business. I didn't know anything about business, so they gave me a business partner, Andy May, and an engineer. We vaulted through the FDA process. We're a Class 1 medical device, and in 2014 became Balance Tracking Systems. Please visit our website if you know anybody who's interested in stuff. Contact me. We've gotten a fair bit of media attention. I won't show the clip, but it's on our website if anybody wants to see it, because we'll get to the science. Some people who are using our system: Michigan State, the Olympic Training Center, LSU. Here's some concussion data that we got from SDSU in a big study we've been doing with them. Baseline data, healthy person. The amount of sway is the squiggly line here. The bigger the squiggle, the more sway they had over 20 seconds. So when they have a post-injury, you can see the squiggle gets bigger, they have bad balance, probably have a concussion. When we looked at the people who had concussions, we found that 6 out of 10 of them showed this pattern of increased sway and worse balance. The best test, the NFL test, only shows in about 3 out of 10. So we're twice as effective at showing balance problems as what the NFL does. The other thing we're really interested in now is getting data from all of our users. So we have people using us pretty much all over the place now. They can send us their data through the software by just clicking a button. I've been gathering that data in terms of athletes. I now have over 3,000 athletes in my database, and we're very interested in basically getting rid of baselining. So the model in athletics is you have to baseline everybody when they're healthy so you can compare them after a head injury. If we get enough normative data, we believe you wouldn't need to baseline anybody anymore. We could just rely on norms. Just big time savings. We're also interested in other things. We're interested in fall risk because older adults fall, they break hips. That's a big burden on the system and on the person themselves, not good. So we're trying to get into that market. We're doing a whole bunch of stuff in rehab. I have many collaborators who are helping me get into various clinical conditions. The great thing about balance is anything that goes wrong in the brain or in the muscles or the bone, you can pick it up in body sway. So we're doing some fun populations. And hopefully I'm on time. Do I have more time?
4 seconds.
4 seconds. These are my collaborators and some funding sources. I've had a lot of help along the way, and if anybody in this room feels they can help us, I'm open to having help. In addition to these people, I will recognize the dean of my college, Marilyn Newhoff, who also has been very supportive of this endeavor. That's it.
Okay, we'll take questions, and because we want to record this, you have to wait till I bring you the mic.
I wanted to know how the sway correlates with cognitive measures of decline.
So what people do in the world of cognitive decline is they do what's called a dual task. So they have people stand on the board and then do a cognitive task at the same time, like counting sevens backwards or remembering things. And what you find is that people who have bad cognitive, they either sacrifice their balance, so their balance gets way worse when they're doing a secondary cognitive task, or they focus on the balance and they do really, really bad on the cognitive stuff. So one of those two things suffers. But is there a direct correlation between how smart you are and how good your balance is? No. Was that your question?
No, it was more of there are traditional measures of cognitive decline that psychologists or neuropsychologists give to people. And so if the sway is a good measure of concussion or cognitive decline, then it should correlate well with those other measures. And I wonder what measures you've used to try to do that, or if you have.
We have not.
Okay.
Sounds like a good thing to do.
I have a quick question. How do you get positive controls? Like somebody with a concussion on your board, how do you deal with not having that access?
Sorry? Like, how do we get our concussed people
Right, I
to
would
test
imagine
them?
you'd want to have a control where you have somebody who has a concussion and you could see their fluctuations in the balance. So how do you deal with the fact that that's not available?
Well, we have had concussed athletes that we have been working with here at San Diego State. So every single athlete at San Diego State, there's 500 some odd athletes in sports where concussions are possible. They get tested by our device, and last year we had 18 of them with concussions, and we post-tested all of them and are using that to validate whether we actually can diagnose concussions.
Dan, I think part of the question is, have you used your technique and the other techniques simultaneously so you could compare them? It sounded like that from your talk.
We haven't done that because they've stopped doing the best test here at State because they think it's garbage. But there's published work showing that 3 out of 10 with the traditional BESS approach is what you see in terms of balance problems with concussion, and worse, about 6 out of 10.
Dan, I was just wondering, what are some of the good biomarkers or molecules that you can use for chemical-based detection to complement your product?
Yeah, some people think LPA is interesting, like Roger Sapadini. They haven't found a real reliable biomarker because concussion's such a diffuse injury of the brain, and it's just hard to locate exactly a single biomarker that would do that. You would want, I guess, a spectrum of biomarkers to try and come up with a signature, I think.
So this is a slightly political question, but by chance I met this morning a woman whose husband is one of the coaches for the girls' track team. I imagine you don't get much business as far as concussions go from track and field teams.
We get them from pole vault.
From pole vault. Actually, her husband was a pole vault coach, as it happens. So my question is this. Would you recommend that San Diego, as I understand, there's no men's track and field team here and hasn't been for some time, and that has to do with, I guess, Title IX and that sort of stuff. Mm-hmm. Would you recommend, based on your knowledge of concussions and so on, that we drop football and replace it with track and field for guys?
I'm a University of Michigan football fan, so I don't care what happens to San Diego State's football team.
You don't have tenure, right?
I'm still upset that you guys sent us Brady Hoke, and then you ruined the whole program in Michigan. Now we're rebuilding. But I think it's a very real possibility that football is going to go away at the it might stay in the NFL, but I mean, Pop Warner, for example, has seen a massive change in the number of people who are signing up to play, for example, and that's just going to trickle up the system, I believe. I mean, I have a son, and he's going to be, believe it or not, he's adopted, so he's going to be like 6'6", 6'7", according to the charts, and like, I'm not going to let him play football. Just not, unless he wants to be a kicker. Because I see these brain slides, and I see enough that, nah.
If nobody goes into football, you're going to lose all of your business space here.
There's plenty of head injuries other ways, and we like older adults as the big final market because everybody's getting old. This is an old population, including myself. I'm getting older every day.
Did we have another question out here? Okay.
Just curious on how you're doing the measurements. Are you measuring the amplitude of the sway, or are you measuring the frequency at which the sway is occurring, or is it both factors?
So there's a billion ways you can break down the sway signal. We do the most simple thing, we believe, which is just take the total length of it. So we have those squiggly lines I showed. If you just took the first point and the last point and just stretched it out like a piece of spaghetti, we just measure the length, the total length of the sway. But there's lots of ways. You can do frequency analyses, and it probably would be interesting to see if any of those other measures are better classifiers of concussion. Some people believe approximate entropy, which is a really hot new metric for balance, is a way to go.
I know most of us, including myself, are interested in the science, but I want to go to your other part, the business side. How's the business doing?
It's a good question. We're still alive. So we launched over a year ago now. We're making enough money to keep growing, but it's slow until we reach that tipping point, I believe.
What are the barriers?
Right now, the biggest barrier is resources in terms of marketing, advertising, and sales. We don't have money to pour into that. But by staying really small, we're able to get by on, selling 4 or 5 a month for now.
Thanks very much, Stanley. Somebody has said, that microphone's not working, but it's not meant to project in this room. This is a small room. It's meant to be recorded. But if you ask a question like this without using the mic, it's the long, big club, and we can wang you on the head with it, too. So our next speaker is Amy Schmitz Weiss from the School of Journalism and Media Studies from yet another college, from PSFA. And she's going to tell you about some of her work that really transcends important journalism studies in different colleges.
Thank you so much. Good evening, everybody. Thank you to Stanley so much for this invitation today. I am honored to be here with you all. So I wanted to give you a story. Imagine, if you will, you're at your local playground with your child and you're pushing them on the swing, and all of a sudden you start to see a whole row of police cars zoom by, and then a few minutes later you see an ambulance that goes by, and you start to wonder what's going on, and then in another instance another police car goes by. Within that moment, you've got your smartphone with you, and all of a sudden you get a text alert from the local news organization that has told you that there's a police standoff 2 blocks away from the playground where you're at. So you immediately start to think, what am I going to do in this moment? So you start to take your child off the swing and head home. Another story to think about. You are interested in food. Perhaps you're a foodie in this room, and you have a great sense of different kinds of restaurants you want to explore here in San Diego. So for the evening, you want to go out tonight and have a restaurant adventure. So you take out your mobile news app and you look at your favorite reviewer for restaurants here in San Diego, and you decide to plop into the mobile news app restaurants that are within 2 miles of where you're at that moment. Up comes 10 options that you can take a look at. You pick one of those and you go out and have a fabulous dinner and cuisine. And then another story to think about. Say you have a friend that is very close to buying a house here in San Diego. They're at the point where they want to put an offer down, they're certain about it. So they start to go over to the area in the neighborhood where the house is being sold. And they're walking around the neighborhood, checking out what's there, what's nearby in terms of playgrounds, libraries, and so forth. And they whip out their mobile news app, the local news organization, and they put in the location of where they are at that moment, about a block away from the house that they're going to buy. Up comes 10 different stories about drug trafficking that's happened in the neighborhood, break-ins, as well as burglaries. As soon as your friend sees this in the mobile news app, they decide to make that decision that maybe this house isn't meant to be, and decide to pull out of doing that offer. What do all 3 of these stories have in common? Well, the idea of taking news content and geotagging it to your immediate vicinity. In order for this to happen and to come together, there's 2 things that have to happen. A news organization needs to have their content geotagged right before they publish that story, down to the exact address, neighborhood, block, so forth. Second, the news consumer needs to have on their smartphone location enabled, as well as a mobile news app to access this information and get text alerts. All of these tie into a concept that I've been working on for several years now called spatial journalism. We are now at a unique point in time where information and geographic space are coming together in new ways than ever before through this little device, or big device if you have the iPhone 6. It's a bigger size. And as a result, it allows for us to have information about the spaces and places that we go through every day and have information that helps us to make decisions. So as a result of looking at this phenomena for the past few years and deciding exactly where is this going, I've come up with a concept called spatial journalism. It's an emergent kind of journalism that allows for us to take the idea of space, place, and location, whether it's physical, augmented, or in many cases virtual, and allow us to think about journalism practices and the actual process in a different way. It breaks it down into 4 key areas in the framework as you see here. 1, it allows for us to understand our news consumption and behaviors in a different way. Based on where we are in that immediate moment in time, what kind of news and information do we want? Is it a restaurant review? Is it about crime in the area for buying a house? Or is it tied to finding out a good place to get food for dinner? Second, tying into the news editorial decisions and roles that reporters make in terms of what to cover in their communities. And how are they making those decisions of what to cover? What is newsworthy in those moments based on space and place? And third, looking at specifically how this information is curated, aggregated, and provided out to the community. We have a powerful opportunity at this time to take the information that's out there from various different kinds of news sources and be able to geotag it to see exactly how are all these different kinds of news stories coming together. And lastly, it also helps us to have a better understanding of our communities, a specific kind of meaning that we give to the places that we live, work, and play, by looking at the spaces and places in that regard. So how does this come to be? Well, I had an opportunity this past year to have a $35,000 grant I received from the Online News Association that allowed for me to explore air quality here in San Diego. It was a collaborative project that I did here with the College of Sciences, Department of Geological Sciences, along with our students in the journalism program and a local news organization named iNewsSource, where we had the students build these little open source electronic air sensors and deploy them throughout the communities of San Diego this past spring. As a result, they were able to have the sensors collect data in real time and map it as you see here. In particular, they were able to gather data points from different observation points in time and to provide stories about air quality in those specific areas, as well as get perceptions from the community on what air quality means to them in their specific neighborhoods. I'm happy to tell you a little bit more details about this after our talk today if you'd like to have more information. But it's just one example of showing how we can take spaces and places that have meaning to us and apply different aspects of news and information, particularly in this case of looking at environmental reporting, of looking at the impact of air quality and its impact on the public. So just to wrap up, this kind of concept of spatial journalism gives us a new lens by which to understand the stories that are being told in our community. It allows for us to identify the gaps, those areas that are being misrepresented or not covered at all. And lastly, it allows for us to have an idea about the implications of the different kinds of tools that are being used for geolocation, like Google Maps, or Weiss, for instance, or Foursquare, and the power of those tools, but also the disadvantages and challenges of those tools and what impact it has on the public. Thank you so much. I'm happy to take any questions.
So Amy did a great job, but allowed a little extra time for questions. So if you guys could have questions, have forth.
Amy, it looked like on that quad chart, you were sort of identifying some things that are special about place-based journalism instead of just sort of place-based information, like I'm here and these restaurants are close to me. Do you mind, can you elaborate a little bit on what you think is special about the journalism piece of having those types of informations in terms of engagement with your community and, you know, the curation of the information and what's true and what's not true from your perspective?
Sure. Yeah, excellent question. Well, when we look at news content itself, it's the process of the journalist that goes through and vets that information. They go through the process of gathering information from multiple sources, verifying that information, providing an accurate and overall picture about that particular location or place. Just to have information in general, you may not get that full picture. You may not get that full representation of that particular location. To get just one particular viewpoint about a restaurant may not be as helpful in terms of a particular restaurant reviewer from the local news organization that's been to 100 different restaurants and knows the ins and outs of what good cuisine is, health inspections, so that you don't get sick in the process. And so that journalistic content is important because it provides that fourth estate, that ability for the public to know that the information that they're getting in these geotagged situations has been vetted, that they can rely on it, that it's accurate, and not just somebody throwing them information. So I think in that regard, it allows for that in that particular moment in time for the person to have the most accurate, complete, and vetted information in that moment.
Hi, very neat approach to the news gathering and being able to see information. I'm curious though, is there any concern that you may accidentally be building a geofence around the user and only seeing news where they go and missing out on things outside and further discovery, or is that a concern at all?
Yeah, that's a great point, and it's a part of this overall picture of asking those questions too, because depending on where that person is in that immediate vicinity, are they only going to be filtering to that particular 2 miles or 5 blocks and getting only that information? And I think it shows in many ways the challenge that we have with working with this kind of model. There's a company called breaking news. They created a mobile news app earlier this year that allows for you to get those alerts, and they define it more to a region or a city in some cases to allow, I think, for some of those variations where you might have some of that aspect of geofencing. But I do think it's another important part, because in that way, that filtering can also reduce the specific information a person gets at that moment.
I'm just curious, what kind of things can you measure with your portable device, and then what kind of spatial resolution do you have? How many of these per square meter, for example?
Okay. That's a good question. In particular, of looking at the data that can be captured, I think we're right at the beginning stages of this. I've been starting to look at different companies that are venturing in this area, as well as starting to look at doing some of my own research in this area, collecting the data. And I think we can look at information points down to specific kinds of content. We can look at specific users that are accessing that information as well as the specific ways in which people identify location, perhaps by cross streets or specific address or by neighborhood as well. And so I think all of those could be analyzed in textual analysis, content analysis, even doing a survey as well to get people's perceptions on what they think about this kind of experience. In terms of the spatial resolution, I think that in time we can have even a better sense of how people identify with the spaces around them in terms of looking at it from that point of view. I don't think we're at that point yet where people think in that mindset, but I think we're going to be getting there very soon as more and more people use their device for this kind of information discovery.
Amy, I think of journalism not only as providing information, but providing education and proper response. And in the case that you described where there's an emergency response, that is something that people really need kind of on-the-spot education about how to respond. Is that also a part of what you're doing? And if so, could you comment on that part of the process?
Yeah, I mean, I think a big part to this too, aside from looking at it from the perspective of environmental reporting, we could look at it from the standpoint of helping in a disaster and allowing for the news organization to partner with specific local, regional, state agencies that could help them to mobilize people in those moments. So if they know, I'm sure many of you have seen that the USGS has an earthquake app now that gives you a warning a few seconds before it's going to happen. But I think for news organizations to do more of that partnering with those kinds of agencies in those moments when that geotagged content goes out will really show the power of allowing for these two entities to provide that educational role, as well as help people in a moment of disaster, hopefully save lives in the process.
So do you ever think about it going the other direction? So if people are voluntarily giving up their location to news agencies, could you ever use them as a way to identify sources of information for the reporter, where you could reach out to them saying, look, we know you were there. What are you seeing? What can you tell us? Have people tried to do that yet?
Yes, they have actually. When the Boston Marathon bombing happened a couple years ago, several reporters went to Foursquare at that point to identify where people were within minutes after it had happened, and started to look at the vicinity of blocks where people were, and started to reach out to them through Foursquare to see if they would be able to give them via Twitter or through phone reports of what had happened, if they were willing to talk to the reporter. So I think having more of that kind of opportunity in those kinds of moments, I think, can be important if the citizen is open and willing to speaking with the press about it.
I think we have time for another question. If you, we have one over here.
Thank you. This is really terrific and visionary and forward-looking, but could you address a little bit about the subjectivity of this process, both in terms of news gathering as well as, choosing, selection, reporting? So, for instance, break-in an affluent neighborhood might be a different story than in a neighborhood which is riddled with, similar events. So to what degree is it going to increase blindness, or perhaps to such events, or disparity between neighborhoods and communities, you see?
Yeah, it's an excellent question, because I do think there can be the potential for that level of subjectivity to come in based on what the news organization, as well as the reporters, decide, as I was showing you in that one part of the framework, of what they think the community is and what it's represented by, as well as where they put their efforts. And so I think we may start to find that there will be smaller news organizations that will focus on a more hyper-local level to get to some of those aspects versus trying to be the big behemoth news organization to cover every area, because they just won't have the resources to do so. So I think we'll start to see some more of these small hyper-local entities that will try and get to those different neighborhoods across the city, for instance, to be able to try and capture that information. But at the same time, this question always comes in my head, is I think if something is not on a Google map, does it mean it doesn't exist? We often just take Google Maps for granted and think that every location, every street that's on there is finite and it actually exists, when there may actually be a business that's not on Google Maps that does exist, that's been around for decades, for instance. And so I think in these moments it's important for us as academics to study this, because it has the potential and implications for the future for people not to be aware of this because of subjectivity decisions that are being made in these technology companies, where citizens as well as the journalists are not being a part of those discussions. So it's a great question.
Thank you, Amy.
Thank you.
And now, like Monty Python, now for something completely different, we have Tom Huxford today from the Department of Chemistry, who will be explaining to us the joys of X-ray crystallography and its utility for understanding novel compounds.
All right. I'm going to try to teach you a little bit of chemistry to give some context to what I think is some pretty neat observations we've made in the last couple of years about an enzyme that we love and we think is going to enable us to be able to come up with some novel approaches to treating disease. So I'm going to present this talk on novel structure-based strategy for inhibiting IKK, which will mean something to you in very short order. I'm going to start with kinases. Kinases are a class of enzyme that catalyze a very specific chemical reaction. They take a phosphate group off of a very well-known molecule, ATP. Anyone who took high school biology remembers ATP, the energy currency of the cell, the high energy compound that every living thing makes in order to be able to stay alive. So kinases are enzymes that will take a phosphate group off of ATP and transfer it to some target small molecule. And usually the purpose of that is to raise the energy of the small molecule so that subsequent chemical reactions can take place without too much trouble. They can just charge right ahead. A class of kinases known as the protein kinases does the same chemistry, but instead of putting the phosphate on a little tiny small molecule, it puts the phosphate on great big protein factors. And to place something as small as a phosphate onto an enormous protein seems like it wouldn't have very much consequence. And the fact is, it's by far and away the most important modification that happens to proteins to control things like metabolism in our cell division, immune responses, and expression of the genes that the cell needs to be able to react to a particular environment in the appropriate way. So the attachment of phosphate to proteins through these enzymes, protein kinases, is something that if you are not familiar with it, something I hope you go home thinking about, because that's a huge thing. It's so important, in fact, it was awarded a Nobel Prize to Fisher and Krebs. This is a great photo, moments after they were told that they had won the Nobel Prize, both of these at the University of Washington in 1992, for their discoveries concerning reversible phosphorylation. That's taking that phosphate group off of ATP and putting it onto proteins. So in the years before and since those original discoveries, it's very well established now that pharmacological control over those protein kinases, being able to turn them on and off at the right times, has a huge potential to treat and cure diseases. Some of our very best small molecule drugs to treat cancers like chronic myelogenous leukemia are inhibitors of specific protein kinases, block them, stop them from functioning, and the tumor is cured, the patient survives. So There's a lot to be gained by coming up with nice, specific drugs that block protein kinases and keep them from their action. But there's a problem. In the genome of the human, there are approximately 518 of these protein kinases, and every one of them catalyzes the same chemistry. That is, they all recognize the ATP, they all take that last phosphate and put it into a position where it can be attacked and attached to whatever the target is for that kinase. And so if we target the protein kinases through that entry point where the ATP binds and the target gets phosphorylated, we stand to have lots and lots of side effects, and that's usually the case. There are there are a huge number of wonderful inhibitors, small molecule drug-like inhibitors to protein kinases that have been tested in the clinic and in mice and so forth that are ridiculously toxic because they don't only stop the kinase that we want to study, they knock out 250 others. But there are other things that kinases need to be able to do. Kinases need to be able to be turned on and turned off. That's called regulation. So one alternative to targeting the ATP binding site and the ability to transfer the phosphate would be to try to figure out what regulates our kinase of interest and design inhibitors that specifically knock out its ability to be turned on and off. And there's another possible avenue. It's the target specificity. So different kinases attach those phosphates to different targets, and if we can learn enough about what makes a particular kinase go after a particular target, maybe we can come up with a mechanism for blocking that. So those are the big questions that we were addressing. The approach that we take in my lab, I call my lab structural biochemistry lab because we rely heavily, almost entirely, on the production of protein structures, seeing the molecule in action, and then using that as our template upon which to make testable hypotheses and advance the discovery. This is a slide. This doesn't show up very well, and I'm not going to turn off the lights. There's a whole bunch of little numbers there. Those are the 518 kinases in the human genome, and their distance from one another sort of represents their evolutionary conservation, how closely related they are to one another. I'm going to be looking at a kinase that's right about here in this no man's land of kinase space, and it's a kinase that's called IKK. IKK stands for I kappa B kinase. That's not the important thing. The important thing is what it does. It controls the cell's response to potentially toxic stimuli by cranking up a cellular encoded survival response. It allows the cell to start turning on genes that normally wouldn't be turned on, but in response to the perceived toxic threat that's being presented to the cell, it's going to turn on those genes and by thus doing promote survival, fighting off the infection or whatever it is that caused the insult. It could be UV light, it could be cigarette smoke, all sorts of different potentially toxic things will turn on this cell survival response. And the key mediator is this kinase, this protein kinase called IKK. And there's some really nice links, nice in the sense that they're beautiful experiments. I don't think it's nice at all because it causes cancer. But the beautiful experiments that show the role of this IKK are that when we take cancer cells, we typically, not in every tumor, but in most of the tumors that have been tested, we typically see that there's a lot of this activity of this kinase in the cancer cells, which is causing them to promote survival. And when we knock down that kinase activity and then give it the chemotherapeutic drug, we can kill the cells very easily because they're not promoting their own cell survival. And even more interesting, I think, in beautiful mouse studies where we take mice that are predisposed to get different types of cancers, we can knock down the IKK kinase activity and see that they don't spontaneously produce those tumors and that they produce much smaller tumors and they're easier to treat. So let's come up with ways to do this. So using our structural approaches, which was the X-ray crystallography that Dr. Welter referred to, this took a lot of years, too many, frankly. We were able to come up finally with our first glimpse at the human I kappa B kinase and its active conformation, and this is what we learned. This sort of salmon-colored portion right there, this is the big spaghetti string structure, is one way of representing an enormous molecule, a protein molecule. And this salmon-colored one right there is the part that does the kinasing. It's the part that puts the phosphates on there. And then there are two other sort of structurally folded domains. This blue one right here is really interesting to us. We had no idea that that's what we would see. And the first reason it was interesting to us is because through that blue domain, this blue portion, it's forming a dimer. It's two molecules are coming together. The first thing we decided to do do is let's check and see how important that interface is to the activity of this enzyme. And so we introduced changes that would occur right here at this interface to make it so it couldn't form that dimer anymore. And when we let loose that kind of a protein inside the cell, it couldn't get turned on. We knocked out its ability to be regulated. Now, if we made those same changes and tricked the protein into thinking that it was already on, it did the chemistry just fine. So we weren't blocking its activity, we were blocking its ability to become activated. And it suggests that this interface might be a place that if we had the right small molecule, we could target that and maybe block IKK activation by stopping its regulation. So that was something that came out of that structural study. Likewise, we found 2 other interfaces in our crystal structures that when we made those types of changes, we saw that they would block the activation without stopping the activity. And to quote my good colleague Jeff Gustafson, that's not even what I'm excited about. What I'm really excited about, and I don't know if I have enough time to say this, the kinase domain, we originally did an experiment years ago where we only worked with this salmon portion all by itself, and we had made a very interesting observation that a lot of people I don't think really appreciated. What we found is when you work with this piece all by itself, independent of the rest, it still functions as a kinase. It can find the ATP, it can put phosphates on things, but it loses the ability to recognize its target that turns on the cell survival. Something about the interface between this guy and the yellow and the blue is what is directing that specificity and turning on cell survival. So we engineered a version by cutting off little pieces that functions as a monomer and captures just that interface, and that's the molecule that we're going to be testing now to try to find some small molecules that maybe by interfering right in this interface between these 3 guys will break that thing apart and make it so that our kinase is just fine and happy, but it can't turn on the cell survival response, and maybe we've got a drug that's specific for this kinase alone among all of them. And that's it. So that's the hypothesis. There we go.
Questions for Tom?
Oh, Crystal. Crystal just published some beautiful work on kinases.
This is lovely work, Tom. Thank you. I guess we haven't talked about, or maybe I've forgotten about the dimerization versus you had some multi-oligomerization going on. So are all of those physiologically relevant, or do you know yet?
Okay, physiologically relevant in the sense that, okay, this isn't real physiology, this is biochemistry. Physiologically relevant in the sense that when we introduce mutations at these interfaces that I pointed to, we can modify those proteins in a way that when we introduce them into cells and then activate the cells to have them turn on that kinase and pull them back out of the cells, they're not on. But then we make those same mutations within the context of a kinase that's already turned on, let them loose, pull them back out, they work just fine. So what we're proposing is those interfaces and that oligomer, you used the right word, the oligomer, that building up of that big complex, that's a necessary intermediate in the pathway toward activation. And it's interesting because if you, this is interesting for you, because most kinases become active, it becomes chicken and egg. How does a kinase become active? Well, another kinase has to attach phosphate to it, and it becomes kind of a cascade type thing. This one is one of these ones where one of the kinases where when they looked for the upstream activating factor, they find a whole bunch of different things that can do it, but none of them is absolutely required. And we argued in the paper that's because you don't need anything upstream. This one can do it by itself. By forming that kind of oligomeric complex, it can get the process started, get the ball rolling. And it kind of makes sense for something as fundamental as NF-kappa B dependent cell survival that maybe it would have its own sort of auto primer to get itself on. That's what we argued in the paper. Reviewers bought it. All right.
So taking a way macroscopic view here, coming from the public health clinical side. So question for you is, taking as this ultimately could be potentially used, how tumor specific do you think this compound would be in terms of tolerability for the patient?
The studies that have been done along those lines are things like this. You have a tumor cell line in culture. You treat it with doxorubicin, or you treat it with cisplatin. It'll kill the cells. Then you do the same experiment, but you first block this enzyme using a non-specific kinase inhibitor. You need very, very little of the doxorubicin or the cisplatin to kill it. The idea being that you've crippled the tumor cell from cranking up the cell survival response, and now you've made it much more amenable. So the simplest idea of how you might use a drug that would come out of this kind of a study would be just hand in hand with already existing chemotherapeutics to make them work better at lower titers. So less side effects, because those are really toxic substances. So that would probably be the most direct kind of approach.
Tom, you talked about some complicated biochemistry, and one of the things. I was hoping it was simple. No, I mean I think the whole point of it is that it's complicated. If it was simple, you wouldn't have to do all this stuff. But one of the things that I think the public is always interested in is you say, well, these crystal structures will allow us to develop drugs and go from here to drugs. Could you say something about how you would go from these structures to finding a drug, and what you would imagine the time frame would be before you would even have something like that that might be able to be used in humans?
Yeah, so the typical approach, what I love about structural biology is you see things that you see what if you design the experiment properly, you'll see things that you planned on seeing, but you'll always see things that you didn't plan on seeing. So it sort of raises the bar for everybody. And now we have, I think, a very nice template upon which to try to do things. But to answer your question, the approach that would be taken would be first doing work on the purified enzyme itself, or even just pieces of the enzyme that mediate those interactions, and set up a high throughput screen against billions of compounds that will fish out of those billions of compounds which are the ones that can disrupt that kind of interaction. Now you've got lead compounds. Then you invite your chemist friends along and you do a sort of an iterative process where you test how well they break the thing apart. Then your chemist friends do modifications to those compounds, and you see if you can get them even better, and you do structure-activity relationships. You can do these things over and over again until you come up with compounds that are very potent at doing what you think they want to do. Then you're going to let them loose in some cell-based system, primarily an animal system like mice, which cancer is modeled really well in mice. At that point, people much more experienced and wealthy than probably we are would want to put up money to get them into the way to get things into trials is you start with a phase 1 trial, which is, I joke about, it's like a taste test. You get a bunch of people lined up, you pick a low concentration and a high concentration that you think you'd like to work in, and you prove with an experiment that in that range it doesn't hurt anybody. And so now that's the phase 1 trial. You've done your phase 1 clinical trial, you've got approval from the FDA to work within that range. Now it's time to start bringing in people who are actually sick and see whether, and the time frame for that, if you're working on a tumor type that already has drugs in the clinic, it's going to be really slow. We're talking 5-10 years. If it's something like a pancreatic cancer though, or something where, the last, one of the last very good drugs that was approved for pancreatic cancer this is depressing to even say it, but it didn't improve the rate of outcome at all. It was approved by the FDA solely for quality of life issues. It just made people a little happier during the 6 months that they have to live. I mean, it's really a brutal disease. So if you have something that's got hope in one of these diseases, like pancreatic, it can get fast-tracked through, and in maybe 2 years you could have a compound that was ready to be in the clinic.
So thank you, Tom. I just want to say a comment before we pull Stanley back up here. And that is, when you're an administrator, sometimes you start to get pretty distant from what it was like as faculty. So I really love coming here because one of the things that I capture back is the joy you can feel when he's talking about crystallography or space-based knowledge, and it really is infectious. And so not only is it fun to learn things, it's nice to get that energy back. So I'm very thankful. So earlier this morning I referred to Stanley as a magician in a fundraising development program, and really because of his mystical ability to interact with so many different kinds of people. But one of his other mystical gifts is his ability to summarize very diverse topics. So we're going to turn it over to the magician.
So this is really a great view of our campus. We've had talks from 3 different departments in 3 different colleges on seemingly very, very different types of things, right? But I think there is a real connection between each one of these things, right? In reality, each one of these talks focused on processing information, a very particular type of information, in a discrete and special way. So the first talk really developed a better way to process the information of motion to provide data that had an application that really can improve human health and is much better than the other ways of doing this process. And the effectiveness of this approach is only as good as the information that it provides and the analysis of that information, right? The second talk really focused on location information and the analysis of aggregation of that information in a way that provides a lot of abilities to respond to things, including response to emergencies. And there were a few examples, but I think that there really are probably 500 million responses that are really critical for all of our lives. Responses to earthquakes, responses to a whole slew of other natural disasters. But when it gets down to the nuts and bolts, it's a new way of getting information and a new way of processing that information to change our behaviors into response to these things. And the third talk was using structural information, and I didn't mean it as an insult to Tom when I said complexity. It's because the cell is so complex that to dissect out the type of information that we really need to develop a new drug demands really clever approaches to be able to take these 3-dimensional types of information and convert them into something that really modifies the behavior of the enzyme and the impact on the cell, if you will. So fundamentally, I think there is a close tie-in between all of these different areas, and in each one of these cases, the processing of that information really uses computational approaches that we didn't have so many years ago to be able to come up with this analysis. So it's a great example of how people on distant parts of campuses doing very different things, I think, can have something in common and potentially can really learn from each other in key ways. So before we finish, I want to say that these Discovery Slams happen because of some key people. And so Debbie Brighton in the Research Foundation plays a really key role in advertising and helping identify speakers and in really the recognition of the research that's very exciting across campus. And Steve Welter, in his role as the Vice President for Research, can see what's going on across the entire campus and help bring it to us so that we really can recognize the opportunity to bring these aspects together. And I'm just lucky enough to be able to play Tom Sawyer and get them to work on these things together with me. So anyway, thank you all for coming. Please, if you can, stay around, eat some of this food, and ask questions to the speakers or others. Any food you don't eat, we have to schlep back across campus, so stick around and we'll see you at the next slam. Thanks very much.